Okay, hi everyone. My name is Riley Macdermott. I'm a rising senior at the Benjamin School, and this summer I had the opportunity to work in the light microscopy core, where my project focused on deconvolving high-resolution images. So here at MPFI, we have a light microscopy core consisting of many relatively fast and reliable microscopes. So these microscopes require time, care, and precision to use. So the light microscopy core specializes in imaging on these microscopes to provide scientists and researchers with detailed multicolored images to analyze. The goal of my project specifically was to find a way to take the nicest possible images with the highest resolution using the microscopes that we have here. So this image specifically shows a ferret brain slice with pyramidal neurons in green and axons in red. To begin, it is important to understand that resolution is how close two structures can be while still being distinguishable from each other. When the separation between 2 points is no longer visible, the limit of the resolution has been reached. A lot of the time, scientists are looking to observe tiny structures that might be separated by only tens of nanometers. In this case, microscopes with a high resolution are very helpful. Of the many microscopes we have here, I got to image on 2 of the strongest, which are the STED, which stands for Stimulated Emission Depletion, and the Zeiss 980 AreaScan 2 microscope. The STED is able to take high-resolution images of smaller structures, but it is limited as to what it can image, as larger areas would take a longer time. Imagine using a very fine-tipped paintbrush to paint an entire wall. The results would probably be a near flawless paint job, but it would take far too long. In comparison, the Zeiss 980 AreaScan 2 is easier to use and can take larger images at a faster speed. However, the resolution of these images is limited to 200 nanometers, where the STED can go well beyond that. Using these microscopes, scientists are intending to look at very tiny structures in their samples, sometimes even as small as a singular synaptic connection. So it is important to find a way to take the nicest possible high-resolution images, like what can be taken on the STED, on a relatively fast and easy microscope like the 980. One way to do this is by using deconvolution. Deconvolution is an algorithmic process that is able to improve the resolution of images taken on the 980. It works by determining which signals in an image are relevant and which signals are unnecessary background noise. Eliminating this background noise creates a detailed picture with clear structures, so we are able to observe important tiny connections. Here, there are 4 different methods that we use to deconvolve images, and the main goal of my project was to test them all and determine which one, if any, offered the best results. In order to tell which method actually worked the best, I needed a standard slide to analyze. These Argolite calibration slides are machined, and they have specific patterns laser etched into them. We know exactly what they should look like and what their resolution is. This pattern specifically consists of pairs of lines, each of which are a different exact distance apart. The distance between the pairs gets smaller towards the bottom of the pattern, beginning at 700 nanometers, ending at 100 nanometers, and decreasing by 50 nanometers each line. Since these exact distances are known, by observing the images of these slides and finding where the gap between the lines is no longer visible, I can tell what the resolution of the image is. The resolution can be mapped by a line intensity profile. So these graphs show the intensity of light at each point on this line. The peaks on the graphs are the areas with the most light signal, and the dips are the black spots that the line passes through. So the resolution of the image can be mapped using these peaks, because when 4 peaks turns into 3, there's no longer any visual separation between the lines, meaning that is as far as the image is resolved. However, the deconvolution software can go too far and create an unusable image. So the software works by recognizing an area in an image with a lot of signal. It then finds the brightest spot and moves the surrounding dimmer light into that spot, creating the optimal image. However, sometimes if the strength of the deconvolution is too high, the software can enhance areas of signal that are not really there and create artifacts in an image. So the first image shows the Argolite slide processed with the strength set to standard, and the second image shows the deconvolution strength set to high. So in this case, the high setting took the straight lines and made them appear uneven and dotted, which is not correct. So my project focused on testing each of the different methods of deconvolution and determining which one provides the highest resolution image with the fewest artifacts. In order to do this, I started by imaging the Argolite calibration slides to get a baseline and determine when the software was working the best. I imaged these slides on the Zeiss 980 AirScan 2 microscope, and I used the 20x and the 63x objectives because these are most commonly used here for imaging. So the 20x is able to see a larger field of view, while the 63x is able to zoom in on a particular spot more clearly. These objectives are, after obtaining images of these slides, I applied the deconvolution software and tested each in order to obtain evidence as to which software works the best. After the conclusion was drawn, I imaged certain brain samples on the 980 and applied the deconvolution software to these samples. This is what the Argolite slides looked like before and after deconvolution, and you can see that after the image is deconvolved, the separation of the lines is much more clear. Using these slides, I found which method gives the best resolution in two-dimensional images, and these are how the slides look after being deconvolved with each different method. After testing each, I determined that AirScan joint deconvolution and SVI Huygens deconvolution are capable of achieving a similar resolution of about 300 nanometers. One thing to consider when choosing between these two methods is convenience. While they both work similarly well, Huygens is more difficult to use and access, while AirScan joint deconvolution is built in to the software, the Zen software that we already imaged with here. So while both work well, AirScan joint deconvolution is more convenient on the 980 microscope specifically. Additionally, not only does deconvolving images improve the resolution, but also the overall signal of the image. On the graph on the bottom right, the red represents the signal before deconvolution and the green represents the signal after. And as you can tell, the green is much greater than the red. Now that this conclusion had been reached, I applied the software to images of different brain samples, including nuclear pores, cytoskeleton, and neurons. All the images going forward were taken by me on the 980 microscope. This video shows a pyramidal neuron in the visual cortex labeled with green fluorescent protein. This is an image of a nuclear pore and nuclear pores and cytoskeleton, where the green is labeling tubulin and the red is labeling nuclear pores. In this image, the structures, especially the tubulin, appear much more defined after the software was applied. Here, the joint deconvolution software is applied to the samples of cytoskeleton, where the red once again labels tubulin and the blue labels vimentin. Once again, the tubulin structures appear much more defined after the image has been deconvolved. Lastly, I have a pyramidal neuron in the visual cortex labeled with green fluorescent protein. And the researcher here was specifically looking to observe the dendritic spines, which after the deconvolution are much more detailed and easier to analyze. So the goal of my project from the beginning was to optimize deconvolution so that images taken on a confocal microscope like the 980 could have a higher resolution comparable to that of the STED. After imaging on 2D calibration slides, I was able to determine that AirScan joint deconvolution and Huygens deconvolution had similar resolutions when resolving images. The benefits of joint deconvolution mainly apply to imaging on the 980, as it is able to improve the resolution of these images, it is easy to access, and it does not require paying for a separate license. Huygens, however, is more robust and compatible with microscopes other than the 980, such as the STED and the light sheet. Huygens also showed no evidence of creating artifacts in the images, while joint deconvolution did create artifacts when pushed too far. So now that the softwares have been tested and optimized, scientists and researchers can use deconvolution to enhance their images for more detailed analysis. The next step with this project would be testing both AirScan joint deconvolution and SVI Huygens deconvolution on larger samples to determine whether or not one method is faster than the other and thereby more efficient. So for this project, I'd like to thank Nikolai, Katie, and Alana in the light microscopy core for teaching me everything I learned this summer, and Ilaria and Erica for overseeing the internship and making it a lot of fun, and the MPFI supporters for making this opportunity possible. Thank you.
I think we all agree that you kill it in the light microscopy core. Questions for Riley?
Thank you. On one of your first slides, you showed that the size microscope had a resolution of about 200 nanometers. However, the pictures at the end where you showed the results, they were 300 nanometer and more. So you couldn't get to the limit of 200?
So the question was that in the first slide, I said that the Zeiss microscope can get to a resolution of 200 nanometers, but in the images I showed, it was 300 nanometers and more for the scale bar. The answer to that, we can get to that resolution using deconvolution, but the specific samples we imaged did not need to go that far. But we have more pictures of samples, and you can talk to me and my mentor after about how far exactly we can go with that.
Any other question for Riley?
You mentioned testing these images in 2D, and I was wondering if this was also possible in 3D.
So the question was if this deconvolution was also possible on three-dimensional images. And for my project, I did test three-dimensional. So the Argolite slides also have a pattern that is a sphere, and it's able to test in 3 dimensions. And we had the same results where the two methods of deconvolutions worked similarly well to improve the signal and the resolution of the images.
Hey, Riley.
Hi.
Thanks.
Nicola likes to
I do.
get the curve,
But-
like look at Alana, she knows.
Yes, I know. I like to torture everyone. No, thanks so much for your work, and I'm really excited to see what all the researchers are going to be doing with what you've basically now helped everyone to use. I do have a question though. A lot of researchers here, everyone's going to turn in their samples and try this because obviously it's going to make their images look nicer. Some people have truly huge, entire neuron branches and everything in 3D with high resolution. Do you have any idea if there's going to be any problems using such huge data sets when try to deconvolve it? Have you noticed anything as the images get larger and larger, or do you think that's not going to be an issue?
So the question was whether or not having a super large sample to image would cause any issues. And as far as my experience has taught me, the largest image I used took about 16 hours to image, and it's actually this neuron here. And we didn't run into any problems. We just left the software running overnight, and in the morning it produced this pretty detailed image. So as far as I've noticed, the larger the sample hasn't given us any problems.
Do you happen to have the before picture of that wonderful one?
I do not have the before picture, not on these slides, sorry.
Hi, Riley.
Hi.
Are you able to tell me when you would use the 980 and the deconvolution software on the 980 versus switching over to the STED and potentially also using the Huygens or other deconvolution on the STED?
Yes, so the question was when would you use the 980 versus the STED? And as far as I know, the STED is able to take the resolution of images without any deconvolution software applied below 200 nanometers. And then the 980 cannot, there's a resolution limit on the 980, so the 980 cannot image anything smaller than 200 nanometers. So if you have a sample smaller than that is when you would need to use the STED.
Any other question?
I don't want to say a question. I just want to comment on the neuron because I had exactly that with the deconvolved on my big screen downstairs, just looking at it. And the fascinating thing is you can just keep on zooming in, and all these details start to pop out, and you can actually look at these individual tiny things and they look amazing. So that's where you can really see all these details, and it's absolutely fascinating. And I spent way too much time looking at that.
Thank you so much, Riley.
Thank you.